Water for injection, or WFI, is the foundational utility and the primary lifeblood of sterile drug formulation. The stakes for this fluid are absolute. Even microscopic microbial or endotoxin contamination represents a systemic failure that can compromise entire batches of injectable drugs and harm patients. The engineering challenge lies in the scale. You are attempting to maintain absolute microscopic sterility across thousands of liters of constantly moving fluid. A common misconception is that this purity is guaranteed by simply testing a sample of the water at the end of the line, right before it enters the glass vial. But end-stage testing is far too late. In pharmaceutical manufacturing, sterility must be physically engineered into the architecture of the system itself, long before a drop of water is produced. A compliant WFI system is a physical manifestation of strict quality by design principles, where every pipe and pump serves a specific preventative function. This is the central document that dictates the physical build of the WFI system, the Quality Assurance Risk Assessment. The sole purpose of this assessment is to systematically identify every possible point of failure in the generation, storage, and distribution of the water. The process separates generic operational risks, like a pump running inefficiently, from good manufacturing practice, or GMP risks. GMP risks are specific hazards that directly or indirectly impact the final quality of the product, and by extension, patient safety. The central problem for the quality assurance engineer is translating these abstract GMP hazards into concrete hardware requirements that can be physically built, installed, and tested. To solve this, QA teams rely on a rigorous qualitative risk calculus that removes human subjectivity from the engineering process. We calculate this risk using two specific variables: the likelihood that an event will occur and the severity of its consequence. This table breaks down the severity scale. Level 1, minor, means no direct impact on product quality. Level 2, moderate, might indirectly affect quality. But Level 3 is the critical threshold, major. These failures directly impact product quality, like contamination, causing regulatory noncompliance. This 3x3 matrix multiplies that severity of consequence against the likelihood of it happening. Notice the top right quadrant. Any Level 3 major consequence multiplied by even a Level 2 possible likelihood automatically triggers a high-risk rating. A high-risk rating mandates immediate action plans, clear mitigation strategies, and physical engineering interventions. This matrix acts as a ruthless filter. Any hazard that lands in the red zone dictates that a physical piece of hardware must be added to the facility design. Let's apply this matrix to the physical process, starting where purified feed water enters the multi-effect distillation still. This line item logs a major GMP risk. If the feed water flows into the still too fast, incomplete evaporation occurs. Incomplete evaporation allows dangerous endotoxins to bypass the distillation columns and carry over into the final product. Because of the severity of endotoxin contamination, the matrix flags this strictly as a high risk, requiring a permanent mechanical intervention. We cannot rely on humans to monitor this flow rate. The design must include a proportional integral derivative, or PID valve, on the inlet line to automatically control the flow relative to the plant steam pressure. Level sensors must also be installed on the first and last distillation columns to trigger automated dumping if water levels get too high. The next distinct hazard we have to engineer for is temperature. As detailed in row 8, WFI must be maintained above 80 degrees Celsius to ensure strict microbiological control. If the WFI gets too hot, the water just boils. That is an operational resource loss, but it remains a low-quality risk. If the temperature drops below 80 degrees, it becomes a high-risk GMP failure, as microbes can survive and multiply. The precise hardware demanded by this temperature risk is a temperature sensor cum transmitter installed directly at the system outlet. If that sensor detects a sub-80 degree drop, it triggers an automated valve to dump the WFI directly to the drain, physically protecting the rest of the system. The complex array of sensors and dump valves on a distillation still are the direct physical footprint of the 3x3 risk matrix. We follow the newly generated WFI as it moves from the distillation columns into the main storage tank and the distribution loop. Here, we encounter the most insidious enemy of sterile fluid distribution, biofilm formation. Biofilms thrive in static, cooling environments. Any stagnant water in a storage tank is immediately classified as a critical GMP risk. The system cannot just sit. It must feature a discharge pump to force the WFI out of the tank and into a continuous circulation loop. But you also have to protect the internal surface of the tank itself. You must keep the walls wet and hot, even when the water level drops. The engineering required here is a sanitary spray ball with a 360-degree spray pattern installed at the return line inside the tank for continuous wetting. The water level in the tank presents a dual threat. Running dry destroys the discharge pumps, while overflowing wastes massive amounts of pure water. The instrument required to solve this is a rope-type capacitance level transmitter. This transmitter uses 4 strict set points: high-high, high, low, and low-low. These communicate directly with the generation still's start and stop logic, automatically halting production before an overflow occurs. Continuous dynamic movement, 360-degree spray balls, and capacitance probes are engineered solely to drive the high GMP risk of microbial growth down to an acceptable state. Designing the hardware is only half the process. In pharma, a risk mitigation strategy is legally invalid until it is proven to work under real-world conditions. This is the function of the final column of the risk assessment table, the test document. This column bridges the gap between theoretical engineering and operational reality. Specific physical components are tied directly to specific validation protocols, starting with IQ or installation qualification. IQ is the physical audit that proves the specified hardware, like the capacitance probe or the PID valve, is actually installed in the facility exactly as it was drawn on the blueprints. Next is OQ, or operational qualification. This proves that the hardware behaves correctly under stress, verifying that the auto-dump valve actually fires open the exact millisecond the conductivity spikes. Finally, PQ, or performance qualification, proves the entire integrated system can hold temperature and maintain sterility over long periods of continuous commercial operation. Only when the IQ, OQ, and PQ documents are physically signed off does the status of the risk assessment finally change to closed. Every inch of steel, every logic gate, and every valve is tethered directly to a line item on a QA spreadsheet. In pharmaceutical engineering, risk assessment is the structural blueprint of the physical facility itself.